Welcome. We're at the 78th Cold Spring Harbor Symposium on Quantitative Biology. I'm Laurie Dempsey, Senior Editor of Nature Immunology, and with me is Harinder Singh, scientist at Genentech, and as I just found out, that you're also going to be at moving to the Children's Hospital in Cincinnati to head up a program.
Right.
Welcome.
Thank you very much, Laurie. Yeah, I'm actually quite excited about this new venture. I'm going to be starting in the fall as the director of the Division of Immunology at Children's, and also setting up a new center for systems immunology there.
That sounds exciting. So one of the things that I want to get into a discussion with you about is that you've had a long-standing interest in developmental fate decisions, and especially using the hematopoietic system as a model system to study. And I'm going to try to get a historical perspective from you, because you've actually seen quite a change of how we've approached problems in this area, and just tell us a little bit about, from your perspective, some of the changes you've seen, like in the past decade.
Sure. Actually, I wanted to step back and go back even further in time, because it's been just amazing to be a participant in what I think has been tremendous advances in this arena. Actually, and I'll roll back the clock to when I was a postdoc at MIT. And actually, I went to MIT to work on basic mechanisms of the control of transcription initiation.
And you were with Phil Sharp. Yeah.
But when I arrived there, I'd actually become intellectually interested in trying to analyze the developmental control of transcription. And I talked to Phil about doing that, and he said, well, at the time, we would consider one of two gene systems to work on, either the globins, and therefore red cell development, or immunoglobulins, and therefore B lymphocyte development. And he said, well, but given David Baltimore here, a close friend and colleague, and all of the work that they're already doing in this arena, it would make sense to strike up a collaboration with them.
Good advice.
And he did, and I have David and all of the many talented people in David's lab to thank for bringing me into the immune system. And at that point, we began by just trying to identify biochemically transcription factors that would be implicated in controlling the expression of immunoglobulin genes, and thinking that those would in turn lead us to the regulators, or many of them might be the regulators that control cell fate choice within the system.
And at this time, it was actually a black box, correct? That we knew the sequence yet of the entire immunoglobulin loci, or was that?
We know at the time, so we knew what the cis elements, some of the key cis elements were. The promoters of these genes had been analyzed, and a key regulatory element had been identified. And of course, the enhancers, the one in the heavy chain locus, and then the intronic enhancer in the kappa light chain locus, had been described, but just a few years before we began. And then the idea was to take elements from these key regulatory sequences in these genes and use them to find the relevant regulators, the transcription factors. And we were successful at doing this, and in fact, I also developed a method which allowed us to clone the genes encoding some of these regulatory proteins. And at that point, as I was transitioning to start up my own lab at the University of Chicago, my view was that we now needed to turn to the biological analysis of the functions of these transcription factors that had been identified.
Relevance question.
Exactly. And to sort out amongst the ones that had been identified and characterized, which ones were regulating key cell fate decisions. Those were the ones that I was particularly attracted to. And so during those first 5 years, actually, one of the regulatory proteins we fortunately focused on was this ETS family member PU1, which turned out to be extremely important in controlling the development of innate and adaptive cells of the immune system, and regulating several different cell fate
not just the B cells, but many myeloid cells and
So even though I was attracted to the immune system from the standpoint of analyzing B cell development, sort of as the model lineage, But the work on PU1 went well beyond that,
Right.
and so it opened up looking at cell-fate choice at an earlier point in the developmental system. That is cell-fate choice that governed the generation of innate versus adaptive cells, or within the innate system as well.
Right.
Yeah.
Right.
And so that's been a tremendous amount of fun over the years. You asked how has the field evolved. Well, I think, you know, we've gone from not knowing any of the components to, I think, having now a very good handle on the major players that are regulating the development of B cells, of T cells, of macrophages, dendritic cells, neutrophils.
More or less, not nearly complete, but close to complete parts list?
Yes, I would not venture to say that we have the complete list by any means, and I think we're dealing with obviously complex circuits, but I think it's fair to say that we have identified, genetically analyzed, and I mean of course the larger community, some of the key players involved in the development of each and every one of these lineages.
Right.
And not only that, but we have a pretty good handle now on the connectivity, the crosstalk between these regulatory proteins, and the crosstalk is sort of at the heart of cell-fate choice.
Right. Because you've talked about regulatory networks, and I remember coming to some of these immunology meetings 10 years ago, and everybody had their favorite gene, their favorite transcription factors. Matter of fact, they dubbed them master transcription factors.
Yes.
And who was really responsible for a given decision? It had to be this player.
Right.
But now we're actually evolved into, it's actually a combinatorial, and in the context of you could have a certain fate, but if there's a different context, then you can actually go down a different pathway.
Right.
And I'd like to get your thoughts on both the evolution of this and then some of the newer findings in terms of how we're putting this together in a conceptual framework.
Right. I think this has been a central and an extremely important issue, and it's also one that I think has driven the evolution of this area. There's no doubt that 15 years ago, as these regulatory proteins were being identified and their functions pinned down, as a reductionist, as all of us, we decided, okay, the one component that we're working on is key, is sort of the master player in controlling the development and the functioning of a given lineage. I think, though, we all of us knew just from the working on the control of gene expression, particularly in mammalian cells, that regulatory sequences are complex. A given regulatory element in a gene is complex. It is seeing multiple inputs, so we already knew about combinations of transcription factors that were needed to activate a given promoter or activate a given enhancer. So I think that clue already said that it wouldn't be as simple as a single regulator accounting for the complex pattern of gene activity that in a given cell type, but it had to be combinatorics.
Right.
And then, of course, the realization that that when you think about development and you think about the complex patterns of gene expression that are manifest in particular cells, that the circuits that are controlling those patterns necessarily involve multiple components, and I think it's conceptually more appealing and actually more productive to think about these transcription factors as components of information processing circuits, acting within the context of those circuits then to control, and it's those circuits that control specific patterns of gene expression. Certainly there are dominant components in those circuits, but it's the circuits as a whole that account for that complex behavior. It's not the activity of one protein.
Right, and then you can set up a certain circuit and then have a feed forward and then reinforcement. So,
correct.
So can you tell us a little more about some of these circuits that come up with, we've heard things about commitment and establishment of some of these lineages. So,
Right.
so just some of the factors, and then what I eventually want to get into is some of the work that you're going to be talking about at this meeting,
Right.
and that is getting back to some of these specific ETS factors and the BATF and some of the other players
Sure.
regulating developmental fate.
Right. So I think the commitment in a sense is viewed from the standpoint of circuit design and circuit dynamics really almost invariably boils down to as you pointed out, feed forward and feedback modules. The feed forward modules are used to consolidate a particular state of gene expression, and the feedback modules are often used to inhibit alternative states of gene expression. And I think as we look into these circuits that are being used to regulate cell-fate choice, whether it's in development or in the context of cell-fate choice in the periphery, for example, the diversity of T-cell states, the nature of the, my view is that the basic designs of these circuits, the architectures, are largely conserved. The components are different, but the modules, if you look at the patterns that are being used over and over again in controlling cell-fate choice, have these rules of involving both feed-forward and cross-inhibitory elements in them.
So let's just to perhaps as a final question, talk about the IRF
Sure.
4
Yeah.
and the BATF,
Right.
the proteins that are
Right.
the topic of your current research.
Yeah, so actually the work on PU1, which is an outlier within the ETS superfamily, led us to identify and clone IRF4, which turns out to be an outlier within the IRF family.
Okay.
And I one of the things that I'm going to talk about here is a very interesting generalization that emerges from our work on PU1, IRF4, and now outliers within the AP1 family, the BATF group. And the reason I say they're outliers is that although these three regulatory proteins belong to three distinct superfamilies, in each case what you find is that they have arisen by gene duplication, but these genes now are restricted in their expression to cells of the immune system. So the immune system has co-opted select members of the ETS family, select members of the IRF family, and select members of the AP1 family to carry out functions within them. The other thing that's striking is that the proteins themselves have diverged over evolutionary time, so that their molecular properties are different from the prototypical members of their family. And the proteins have evolved now to selectively interact with one another. So we've characterized PU1 and IRF4 complexes that recognize particular class of sequences, and those are found in lots of different immune cell specific genes. And now recently we've turned up the fact that IRF4 and the related family member 8 can also interact quite specifically with the select members of the AP1 family, the BATFs, recognizing a different class of regulatory elements, and again, controlling a large set of immune-specific genes. So the broader generalization is that the immune system has co-opted these key members within the broader superfamilies, and then diverged the protein structures. And now these proteins are executing very, very specific functions within the context of innate and adaptive cells of the system. And so that's something that I'm going to enlarge on in my talk here.
That sounds great. That sounds great. Thank you.
You're welcome.